Okay. Hello, everyone. My name is Alejandro Mendoza Cantu. I'm a PhD student at the University of Antwerp. And today, I'd like to share some of the work that we have been doing on reference data-driven metabolomics, an approach that helps make untargeted metabolomics experiments more interpretable. But first, I'd like to, for the last year, I've been working with untargeted metabolomics data, and it feels a bit like this. The first time I started to work with this data is just a sea of molecules that probably have some very rich information encoded into them. And in a way, it also feels like looking at a fragmented image. So I believe that this method that I'm going to present today, reference data-driven metabolomics, will help us make pictures of untargeted metabolomics data clearer. So in untargeted metabolomics, we usually end up with thousands of spectra, each one representing a probable molecule in a sample. The problem is that most of these are unknown. The usual approach to make sense of this data is to search these spectra against spectral libraries, trying to match them to known compound standards. Reference data-driven metabolomics has a bit of a different approach. Instead of relying on just compound libraries, we match our unknown or known spectra against a reference dataset that comes from a known biological context, like, for example, foods, microbes, or other kind of specific sample types. So even if we don't know the exact identity of the feature, if a sample consistently matches our reference data, we can start to say something about our samples, right? So all of our reference data also has attached to it rich data. So to do reference data-driven metabolomics, we're here, we need our sample, MSMS data. So this sample, for example, some kind of human, I don't know, some stool sample MSMS data, and our known biological reference MSMS data. Attached to it is a reference metadata that includes, normally, it's good to know, to have an ontological structure attached to it and detailed descriptions of the reference samples. So this way, by matching our experimental features to these references, we can transfer that metadata also to our experimental or study samples. This allows for more powerful downstream analysis, for example, grouping by different ontological levels. So some of the key attributes of a reference dataset, as I mentioned earlier, the reference data should add some kind of biological context to our samples. That means that it includes MSM well-characterized or well-known samples. Also, it comes paired with rich metadata that describes those samples in detail. And finally, this reference dataset should help us answer a specific research question. Depending on the context, different types of metadata will be important. For example, if we're trying to map dietary patterns, having a foods reference dataset with detailed food ontologies will help us see broader or more specific patterns and groups in our data. If we care, for example, if we are finding some kind of process in our data, for example, fermented versus unfermented foods, if we care about the distribution of the different metabolites along different locations, if we, for example, care between single ingredient or complex ingredient foods, we have to tailor our metadata to these kind of questions. And the question of whether we could detect dietary patterns with untargeted metabolomics data was the first question that RDD helped to answer. The first reference dataset used to apply RDD was the Global Foodomics Project dataset, which is a massive, one of the most extensive MSMS food reference datasets available. It has over 3,500 different foods. And what makes it more powerful is that each of those food items in this reference dataset is paired with rich metadata description, and also structure with this ontology. Here are some of the examples of how this reference dataset is organized. We have 2 levels of ontology, one where we have broader groups, and then we can go into more granularity if we traverse to the different levels. Also, we have in the pie charts some of the details that we have for this dataset, a single ingredient or multiple ingredient kind of foods, if it's fermented or no. So this way, we can also filter our analysis to more specific questions we have. And so now that we have a reference dataset, we need to find a way to link our reference dataset and our sample dataset. To do this, we use the GMPS framework to run a classical molecular networking job. And one of the key steps in this workflow is MSMS clustering, where similar spectra are grouped into consensus features or nodes based on their causing similarity of their MS2 fragmentation spectra. So we find the similar features, which then we see which samples come these match features from between our samples and the reference data. And with this information or of the clusters that were done and matched, we start to create the RDD spectral counts table, or reference data spectral counts table, which basically summarizes how many features from each reference match with each of our samples, giving us a clear overview of the overlap between our study samples and our reference samples. We can also enhance, and this is the main point of reference data-driven metabolomics, we can also enhance this spectral counts table matching our metadata that I talked before. For example, our ontology in our food data. This allows to summarize not just which references match, but also how many features match groups in this ontology. And so this example with the Global Foodomics Project dataset is a first application, and it was possible to detect differences in food intake between individuals following a vegan or an omnivore diet. When we look at the data group, for example, at the level 3 of the ontology, we clearly see differences in the intake of dairy, meat, and seafood products between omnivore and vegan groups. Also, if we go into more granular data, performing a PCA on the most granular level of the ontology, we can see here that we can see a clear separation between the individuals from the omnivore and along the first component of the PCA. This suggests that the we obtain through RDD, both aggregated and not, reflect in the chemical compositions of the samples, in this case, driven by diet. And so going back to the picture, at least I hope I have convinced you that now at least one part of the picture is more clearly seen. But now we are extending reference data-driven metabolomics to another use case, which is focusing on microbial reference data. So we built reference data for microbes using MicrobeMast, which is a reference data set made of organisms with over 57,000 MSMS runs. And we selected those with reliable species level metadata. This allowed us to build a clean, taxonomically enriched reference data set of microbial spectra. We then used this reference that we built, and we started to map samples from the American Gut Project to it, linking them to specific microbial references. This way, we can start to infer microbiome profiles directly from mass spectrometry data without sequencing. And so just some brief numbers of the proportion of the shared features between the human and the microbial datasets. The low match rate suggests that RDD selective to the biological domain we are exploring. and the features are biologically relevant and not just a process of spectral overmatching. Here are the total spectral match counts of different microorganisms. We can see strong spectral matches to common gut microbiome microbes, like E. coli, some Bacteroides species, and some Lactobacillus. This is encouraging because it shows that even without the specific compound identifications, minimum signals from key gut species directly from untargeted MSMS data. It supports the idea that RDD can be used for microbiome profiling and comparing unknown features to a curated microbial reference dataset. This is still work in progress, but they are very promising results. To support all of these, we have also worked on the technical improvements of reference data-driven metabolomics. We've developed a full framework in the form of a Python package, along with a web Streamlit application to support fast and flexible analysis using RDD. Diagram. The workflow starts with the reference metadata, the reference and the MSMS data, along with the corresponding metadata. These are processed through GMPS, as explained before, to get the matching clusters. And then our RDD package takes over using that output and metadata to generate the spectral match tables, the ontology level summaries, and a range of different visualizations and analysis. If you'd like to trade yourself, you can scan the QR code or visit the link to launch the app. Just a few examples of how we have developed the Python package. With a couple lines of code, we can get a very meaningful spectral counts table. And also, we have the capacity of also visualizing the results from our analysis. And with this, I'm showing that it's very easy, for example, to replicate the results that I showed before for the dietary pattern. To conclude, reference data can shape what we can learn from untargeted metabolomics. RDD turns curated spectral datasets into rich biological context. We have extended RDD beyond food. It can help us uncover microbial signatures in human MSMS data using taxonomically enriched references. We have developed an open source Python package and a user-friendly app to make RDD analysis more reproducible and more accessible overall. And the more we share, curate, and standardize our reference datasets, the more we can unlock the true potential of untargeted MSMS experiments. Reference data-driven metabolomics is a framework that grows better the more reference data it has. And now, so this fragmented picture that we started with which we started now becomes a clearer and more informative picture of our biological data. Thank you for your attention. I'd like to thank my promoters, Julia Goglitz and Bob Vitremieu, also the ADREM Data Lab in the University of Antwerp, which are all my colleagues, and they're great people to work with. My collaborators of the UCSD, Potomac, South Africa, and the University of California, Riverside, and the FWO for making the resources available for this project to be possible. Thank you.
Thank you, Alejandro. Time for questions.
Thanks for the talk. I'm very curious about the technical side of your clustering algorithm. Do you deal with like within the experiment or across the different projects?
Yeah, it's across different projects. So using a reference data set.
So how do you take in account for different instrumentation, resolution?
Yeah, it's
all those metadata.
Yeah, we don't specifically. It's a classical molecular networking job. So the only In this case, for the clustering, which is the base part of matching the samples, it's a MS-MS cluster and taking into account the cosine similarity of the different spectra, basically. Yeah, but no, like specific instruments or retention time is not taken into account for this.
So there's no extra like embedding or
No,
some of, okay.
no.
Thanks.
Thank you very much for the talk. It was really interesting. I'm going to elaborate a little bit more about what the previous question about the clustering. So, I mean, I struggle to understand, how do you do the clustering in terms of cosine similarity, and why are you choosing cosine similarity, and in which way? Because you're matching a spectra. So if you can elaborate on a little bit of how the cosine similarity works for the clustering, I would appreciate it. Thank you.
Yeah, I mean, we're not developing the specific clustering algorithm, at least not this project. We're using the framework of GMPS, the classical molecular network, which basically takes the MS spectra and finds and does the cosine similarity between pairs of spectra. The basic algorithm is cluster MS-MS. And yeah, basically that's the framework we use to create the matches. It would be interesting also to explore other clustering algorithms to it, for sure. Yeah.
Hi. Final question. You showed the PCA plot,
Yeah.
where you separate the two groups, and it seems to me that one principal component is enough to separate these two groups. Did you look into which molecule separates the two groups best?
No, I have not looked into it, but it would be super interesting, yes, to see the ones that drive the PC1 the most, for sure. Thanks for the suggestion.
All right. Thanks, Alejandro.
Thank you.